excited to kick off 2023 RECOMB-Genetics. We're really glad that everyone can make it out. We're looking forward, Sriram and myself and Amelia who couldn't be here, but we're really excited across all the submissions and everything this year, and so there should be some exciting science to discuss today and chat about. And so I'd like to thank very much our keynote speaker, Dr. Na Tsai. So I've known Na for some time, primarily through CGSI and various other interactions over time. We're thrilled that she could be here today to present her work. So Na focuses on leveraging genetics to understand major depression disorders. And so a lot of the challenges that come with phenotyping with respect to heterogeneity of outcomes and how that complicates the analyses with respect to any sort of genetic or molecular findings. And so amongst many other aspects of that. And so we're thrilled that Na could be here today and share with us her excellent work. So thank you so much.
Hi everyone. Thanks Nick for the introduction and for inviting me to talk at this RECOMB satellite. Very happy to be here. I also should introduce myself. I didn't know that he was going to introduce me. So I'm a PI at Helmholtz Munich, and currently I have a group of 5 people. We study genetics of depression using biobank data, clinical cohorts, and going forward maybe electronic health records as well. And we also use publicly available as well as custom omics data to sort of supplement our findings. Sorry, I don't know how to use this computer. To supplement our findings from genetic data. So this is my first time at RECOMB, and it's nice to see everyone. But I just want to say at the beginning that my work is more on the applied side, meaning I don't normally write statistical models. Rather, I apply existing models to the problems at hand, and often I would also identify problems that necessitate the development of new methods. And I usually do that collaborating with groups that focus more on statistical methods. So when I got the invite from Nick, I asked him, what do you want me to talk about? And he said, since I live in this sort of space between applications and methods, I should talk about how that works. So this talk today hopefully will show you things from the application side, and hopefully this is an interesting perspective for you. So before we start on the genetics, I want to tell you a little bit about the disorder that I'm working on and why it inspired all of the thinking and the kind of problems that I deal with. So I work on genetics of major depressive disorder. It is a very common and debilitating disorder that is firstly poorly diagnosed and also have low treatment efficacy, and we also don't understand the biology behind it. So in terms of public health need, there is a very urgent need to make the situation better. And the hope is to understand the biology of this disease using genetic means so that we could improve this situation. So this is the current clinical diagnostic criteria for depression, and according to the Diagnostic and Statistical Manual of Mental Health Disorders. Now this is in its 5th iteration right now, but really it hasn't changed much over the past 50 years. So what happens if you go into the clinic is that a trained psychiatrist will conduct a structural interview with you. And then what happens is if you have either of the first 2 symptoms for 2 weeks, you then go on to answer the rest of the questions. And if you have a 5 in total in the same 2 weeks, then you qualify as a clinical case of major depressive disorder. The thing is that this is not the only way that you can assess depression. In fact, there are loads and loads of different scales that you can use to measure symptoms and then sort of like score depression. So here I just show the sort of 7 most commonly used ones, and they index a total of 52 symptoms of depression. And you can see that some are indexed only by 1 scale, and some are more in consensus between scales. So why do 9 of these made it into the DSM while the other ones didn't? Now that is mostly shaped by historical forces of opposing viewpoints in the development of psychological theory that one of those competitors died of a Vitex, and that's a real thing. And as a result, their theories didn't make it in the end. So In other words, these symptoms are kind of chosen arbitrarily, and they are not better or worse than each other in terms of indexing depression. So those symptoms that are chosen to be part of the DSM criteria are meant to be indexing depression and are not meant to be exhaustively describing it, or they are not the disorder itself. So in fact, Jaspers in 1913 already figured out there's this issue. And he says that it's the main task of psychiatry to find the natural disease units, which are distinct in principle without overlap from each other, and which have the same symptoms, cause, and physical abnormalities. Now, he said that in 1913. 110 years later, we still haven't managed to achieve that. We still are working with indexes of the disorder rather than the disorder itself. So unlike other diseases, this what he's envisioning isn't specific to psychiatry. Basically, all of medicine wants to do the same thing. The only difference is that for things like diabetes and cancer, we figured out what it is, whereas for the psychiatry, we still haven't. And part of the reason why we haven't figured that out is we've started treating indices of the disorder as the disorder itself, and that's called reification. So what that means in genetics terms is that when we do genetic studies, we often aim to find the genes for depression. However, we have defined it. We've defined depression some way, and then we try to find the genes for depression. And then we may take those genes and then go on to figure out whether they are likely drug targets or not. What we're not doing, if we approached it this way, is looking for ways, and using genetic insights to help improve an imperfect model for depression. So we actually never get any closer to the natural kind than what is already being defined. What we should be doing is epistemic iteration, and why we haven't done it maybe is because it's a much harder thing to do. To do that, we need to ask questions like, are we indexing depression well using the current set of criteria? How do we make it better, and what does better even look like? So in my opinion, these are much harder questions to answer than finding genes in a GWAS, and these are the kind of questions that we hope to ask in a lot of our research. So hopefully later on in this talk, you kind of see my attempts at doing this and where we're headed. So there are two parts to this talk. The first part is talking about how I've been assessing depression operationalizations. Operationalization is just another word for definition in GWAS studies. And the second part is how we use genetics to assess symptom-disease relationships in order to get to disease subtypes. So we'll first talk about depression definitions. At the beginning of the sort of early 2000s, when sort of GWAS first became a thing, there are many small cohorts, each with a few hundred or thousands of cases collected through clinician interviews around the world. And each of them isn't able to give any significant genetic finding. So what happened in 2013-ish is that a group of them have banded together and put together a meta-analysis of 9,000 cases and 9,000 controls from 9 different studies. And as you can see in this Manhattan plot, there's still no significant GWAS finding. So then came the CONVERGE study, which I was involved in, which is a single large cohort of deeply and homogeneously phenotyped depression cases, of only severe recurrent depression cases and also screen controls that don't have any mental health illnesses. And all participants were women, and they have all the same ancestry. So it's the most stringently and homogeneously phenotyped cohort to date that is relatively large in size also. So what we were able to find the first 2 genome-wide significant hits for depression, and we think that the success is due to the fact that it's much more homogeneously collected, and there's much less noise in this data set than the one that I've shown you just now. And in the next few years, there came a period of time where groups started taking on radically different approaches, abandoning diagnostic criteria. All of the 2 studies I've shown you in the previous slides, these 2, they've used the structural interview. They've used the DSM criteria for defining depression. So what happens from 2016 onwards is that people started abandoning it altogether. Okay, let's not do that. That's way too time consuming. Let's start asking people questions. Have you ever been treated or diagnosed with depression? And as a result, the sample sizes grew dramatically over these few years, as well as the number of GWAS hits. So, and then people started putting together some of these cohorts into meta-analysis, and it just grew and grew. So the latest GWAS in depression was done on published was done in 2021, where they've used a combination of depression defined by EHR diagnostic codes, self-reported depression, and depressive symptoms. And they found 178 hits for the electronic health record version and 145 hits for the self-report version. So this strategy of using whatever definition of depression you can put your hands on is really based in this hypothesis that all of these different definitions of depression fall under the same disease liability spectrum. So for example, the most severe depression that you could define using clinical criteria plus impairment, treatment resistant, etc., would be on the high end of that disease liability spectrum. And the ones that are just self-report, maybe they didn't get a diagnosis, maybe they didn't have an electronic health record, they'll be somewhere around here. So people have proposed this continuous model for MDD, and this is the sort of basis for doing all of the different definitions of depression you've seen on the previous slide. And this liability distribution is, of course, also what we use as the sort of liability threshold model in genetic studies, where there's contribution from both genetic and environmental factors. So in other words, what we're assuming is that all of these different definitions of depression, they have the similar genetic contribution and similar environmental contribution. So if we agree with the continuous model, then we're great. We have like hundreds of hits, and we have loads to work with. So firstly, we could find map each locus, and we can identify the causal variants from the bunch of correlated variants under a locus. And there are many methods on this right now. We can do TWAS. We can link the potential causal variants with potential causal genes. What TWAS does is it identify the genes that are likely to be associated with the disease using the genetic variants that are associated and their predicted gene expression levels in different tissues. So that's exactly what the latest GWAS and MDD has done. The bottom-most of this plot shows the usual Manhattan plot of a GWAS, where you have a lot of genetic hits. And then this bit over here shows you those that are fine mapped, the variants that ended up significant in fine mapping. And then finally, they overlapped it with those genes that were significant in TWAS. This is done in a multi-tissue TWAS. You can also do it with a single tissue GWAS, a TWAS, where you can do it individually for, they did it individually for 13 different brain regions, as well as whole blood, with the sort of lowest p-value on this end and the highest p-value on that end. So genes on this end are the ones that are most likely to be associated with depression in the various tissues that you see here. So then we could take these, go to a drug company or to go to experimental labs, ask them to knock it out or whatever, and start the whole process of drug identification. But if you question, that's if you didn't question the model. But if you question the model and ask if all the depression definitions really lie on the same liability spectrum and index the same disease, then very quickly, you realize that they're in fact genetically partly distinct. So this came from the same paper, the latest GWAS on depression. And what they've shown here is genetic correlations between different definitions of depression that they put into their meta-analysis. So you can see that for some of them it's great. That's like genetic correlation of 1, which means that all of their genetic effects are shared. And then some of them like 0.59, like really low. Now for context, why do I say 0.59 is low? The genetic correlation between schizophrenia and bipolar disorder is 0.65. So then, for two things that you're calling depression, if they are lower than the genetic correlation between two diseases that are supposedly different from each other, then that's I think, considered low. Ooh, I don't know what I'm doing. Okay. And I've done some analysis along the same lines using a single cohort, the UK Biobank. And here I've defined 7 different depression definitions in the UK Biobank dataset alone. And I've shown that they have different heritabilities, and their genetic correlations with each other also go from anywhere between 1 to 0.6. And that basically mirrors the data that I've shown you in a previous slide. So is this a problem? What does it mean for all of the GWAS findings that we have from all of these big meta-analyses? Is this necessarily a thing that we need to be worried about? So I'll show you that it is a problem in two ways. The first problem is low specificity. So if I take all of the GWAS findings from the self-report version of depression in UK Biobank, I've got 20 hits here. And I plotted out their effect, the odds ratios on the x-axis. And I've also plotted out the effect sizes at this phenotype here, which is help seeking without depression. This is DSM-based depression, clinical criteria-based depression. And this is neuroticism, personality trait that is not depression. And what you can see is that the pattern of effect sizes are pretty much the same across all four phenotypes. So in other words, these effects are not specific to depression, like they are shared across everything. So then, this is the first problem, low specificity. There's a second problem of low sensitivity. And here I show a twin study which tried to decompose the genetic and environmental contributions to depression defined by clinical criteria, depressive symptoms, and neuroticism, and how much they are explained by this common factor of broadly defined depression, which is very similar to the meta-analysis that we see in GWAS. And what they found is that 65% of the genetics of depression, of the sort of clinically defined depression, is not accounted for by the broadly defined depression common factor. And that means that we potentially be losing out a lot on genetic effects if we were to perform this kind of meta-analysis. We'd not be able to find a lot of the unique effects that are meant to only appear in clinical depression. Okay, so that's all terrible. What should we do then? Should we throw away all of the millions of data points on self-report and start slowly accumulating data through structural interviews? And that would take ages. And that's kind of the reason why those self-report studies were done in the first place. So there are many solutions to that. Like the first one is, let's say straight-up meta-analysis doesn't work. What I've shown you just now is that if I did a straight-up meta-analysis between all of these different definitions, it doesn't give us good specificity. So what about weighted meta-analysis? Maybe we could do something that is a little bit smarter. So basically, for example, if we have depression, well-defined depression as one of the input phenotypes, and we have other phenotypes that are genetically correlated with it at some degree, what we can do is that we do a weighted analysis by weighting the effects of these two phenotypes, weighting them by the genetic correlation with depression. And what you see immediately in this setup is that you need to choose these things, right? Like, and in order to perform this analysis, obviously. And the thing is that what you choose as inputs matters a lot in terms of what power you're going to get and the specificity you're going to get. Here I just show you the power differences between two different kinds of inputs. In one of the MTAG runs, I have lifetime depression, which is the sort of clinically defined depression in UK Biobank, together with family history of depression. And here's another run with lifetime MDD again, with self-report definitions of depression and a bunch of other things. So what you can see is that they have drastically different GWAS power. Here you have 5 hits, there you have 30 hits. And that comes with other different implications and consequences as well. Now we talk about specificity a bit. But from the look of it, you already know that what you choose as inputs for MTAG matters quite a bit. So what if I don't know how to choose and don't want to spend any effort figuring that out? So then we came up with another method, multi-phenotype imputation. This is an off-the-shelf method. We didn't develop this method. It's called self-impute. It's an SVD-based approach where you have a phenotype matrix of a lot of different phenotypes that are depression-related, depression being one of them. And in the UK Biobank, you have a lot of missing values of this column, which is the well-defined depression. And that's the whole problem. You have too low power in this one. And then you have a lot of shallow phenotyping of depression, which is collected in a lot more people. And then you have other mental health measures, etc. And what you do then is you decompose that into factors and some weights, and then you then combine those factors and weights to fill in the values, these missing values. This is one way to do multi-phenotype imputation. There's another way that is developed in Shuram's group, a deep learning-based imputation method that has an autoencoder architecture. And I think Ozzy talked about it in RECOMB last year. So in most cases, this approach does better than self-impute in terms of the imputation R-squares. So it does slightly better at the phenotypic level. So what happens at the genotypic level if we were to do GWAS on these? On the top, I show the results from soft-impute, which is the SVD-based approach. These are the data, the GWAS done on imputed values on the previously missing people. So in these individuals, in 269 individuals, they don't have values for lifetime MDD in the beginning. We've imputed all of them. And we have 13 hits, and this is the result for autocomplete, the autoencoder-based method. And there we have the results for if we combine the observed and imputed values for both approaches. And you can see that they improve GWAS power by a huge amount. So then the question is, well, it seems that both MTAG and imputation approaches do, to varying degrees, well in terms of improving GWAS power. But the question is, how good are these GWAS hits? How good are these effects that we find? Are they relevant to depression? Are we going to be able to use them for anything? So then one of those ways to test that is to ask if those effects, when put together, are they going to be able to help you predict depression in an independent cohort? And that's done with polygenic risk scores, which is basically getting the effect sizes at the genome-wide significant hits, wherever you draw that significance line, and combining them with genotypes in a bunch of people that you want to be predicting depression in, and then asking if the predicted depression from these effects, how well do they compare with the actual phenotype of these individuals. So we've done that various ways. I'm going to show you the cross-validation that we've done in UK Biobank. So this is in-sample 10 times cross-validation prediction. In yellow, we have the observed phenotypes. They don't do very well because there's not that many people, there's not that many samples here, the power is low. And in red, we have the imputed. I don't know how well you see the colors, but in red, we have the imputed phenotypes, and they do much better than the yellow ones. So the observed value is this one. All the red bars are around 2 times or more higher than, in terms of prediction R-squared, than the observed. And MTAG, depending on what you put in, does well as well. Some of them do much better than imputation also. So that's all fantastic. It seems that we're capturing the right effects. They seem to be predicting depression all right. We still have the question of whether this improvement is specific to depression or not. So we've done the prediction in some other phenotypes. So here we're not predicting depression anymore, we're predicting things that are other than depression. So neuroticism score, whether you have a stressful life event recently, what's your Townsend deprivation index, which is a measure of socioeconomic status, and whether you have traumatic events happening in your life. And all of these things, for all of them, the x-axis has stayed the same as the previous plot. So you don't need to remember what the individual things are, but just remember that the trend is the same across all of these different phenotypes that we're trying to predict. So imputation and MTAG not only improves the prediction of depression, but also improves the prediction of all of these other things. So in other words, this improvement isn't specific to depression. Again, is this a problem? Should we necessarily be worrying about it? Not necessarily. The thing is that all of these different phenotypes are genetically correlated with depression in some way. So when something improves the prediction of depression, they would also improve the prediction of these other things. The question is, how much are they improving the prediction of these other things as opposed to depression? You want that to be sort of capped at that genetic correlation with each other. So then seeing this, we thought, okay, well, maybe we could come up with a metric for specificity that is based on the ratio of prediction R-squares, and basically tying that to their genetic correlations. So I can show you how it works in this cartoon plot. So if I have a PRS that is derived from the DSM-based depression in UK Biobank, if I use it to predict itself, and I have this ratio here, the numerator and denominator will be exactly the same, and then you'll be at 100%. If I then use that to predict depressive symptoms, this ratio will be slightly lower because it would predict the symptoms less well than it predicts itself. So then it's somewhere around 60%. And then it goes on and on, depending on what kind of phenotypes I put in. How low this is depends on the genetic correlation between depression and all of these other things that I'm testing. So I can do this for one phenotype and then get a curve, basically, ranking the sort of PRS pleiotropy metric from largest to smallest. And I've done that in around 60 phenotypes. So if you look at real data, the black line is the line that I was talking about just now, derived using PRS from lifetime MDD, the DSM-based depression. And you can see that it goes down relatively quickly. So from 100, and then it sort of like quickly goes down to like 40, 20, et cetera, et cetera. The pink and gray lines are self-reported depression from different cohorts. And you can see that those lines are higher than the black line, which means they are predicting other phenotypes better than the DSM-based depression in most cases. And then that basically shows you how much less specific they are to depression. To make this even clearer, you can take this line minus this line, and then sort of normalize it, and then you get this picture, which is what we call excised PRS pleiotropy. So now the black line is now at 0, and then you get the difference between the pink and gray lines to the black line in this plot here. So what you want out of a specific PRS for depression is to be as close to this black line as possible. You don't want it to be nothing. You don't want it to go straight down to 0. You want it to be as good as this black line or that black line over there. So then let's see how MTAG does. These are 4 different runs of MTAG with different kinds of inputs. And you can see that they do very differently from each other. Some are more specific than others. So this blue line here looks great. It looks like it has really fantastic specificity. And that's the one that is derived using family history measures of depression going into the MTAG run. That line over there, that's the one with all sorts of inputs, including Townsend deprivation index, stress to life events, etc. And while that line, as I've shown you many slides ago, gave you many, many more GWAS hits than this line, it's a lot less specific, so maybe you don't want to use that. For imputation, here are the 2 kinds of lines for the SVD-based imputation and the autoencoder-based. And for the SVD-based, you can see that it's lower than the gray line over here. So it's more specific to clinically defined depression than a self-report depression would do. And that's after incorporating 200-odd phenotypes in its model. So it's a very cheap and sort of low requirement way to improve power and specificity at the same time. You really don't need to know a lot about these phenotypes before throwing them into an imputation matrix and achieving this kind of results. So that's great. We talked about imputation and MTAG in terms of integrating different types of phenotypes and still preserving specificity to depression. So we don't need necessarily to throw away all of those self-report data points. What I've shown you now is how we would test for specificity with DSM-based depression. Remember at the beginning of the talk, I told you DSM isn't very good. And so why are we necessarily wanting this at all? And the thing is, we want to be, the final goal is to improve DSM, like to sort of come out with a better way to index depression. If we don't use the DSM to define anything now, then we really can't hope to improve it. And I'll show you why. So here's probably currently how things look like in, this is kind of model of how it looks like. Let's say these things are symptom networks, and here are some environmental contributions, and then they sort of make up what are psychiatric disorders. And let's say this blue circle here is what we've drawn to encompass depression. And this red circle, let's say, is like bipolar disorder or something. So as you can see, maybe they're not perfectly drawn to encompass the symptom network A, which would be depression, and symptom network B, which would be bipolar disorder. They're drawn imperfectly, there's some overlap, it's a bit unclear. And these boundaries of these circles what the DSM boundaries are. They are the definition of those diseases in the diagnostic manual. And if you drew them like distinctly, then you can see the distinct boundaries between those two diseases. Now, if you start modeling up those boundaries and start including lots of different definitions of depression or bipolar disorder in genetic studies, then basically what you're doing is like making that boundary very unclear. Now suddenly you have way more overlap than before. Then how do you hope to how do we hope to improve definitions of depression? Is to look at this overlap and ask whether the things that are happening in this overlap are etiologically relevant to this side or that side, and then you can then sort of think about where to redraw that boundary. So now with the boundaries being really unclear, how would you hope to even do that? You can't even identify what is in the middle. In terms of genetics, what that means is all of the genetic correlation estimates are going to be a bit off. They're going to be higher than what otherwise it would be. And also, our case GWAS attempts would have a particularly hard time because your case of depression really looks like your case of bipolar disorder. Your case of schizophrenia looks really alike too, so that you can't really identify genetic effects that are distinct between them. So that's why even when DSM criteria is not that great, you still have to use it in order to hope to improve it at some point. So what I said at the beginning was you shouldn't believe it and sort of just use it as it is, but you should use it with a mind to improve it later on. So I've come to the end of part one. In summary, different definitions of depression give different genetics. We need to work with the current index in order to hope to improve it. And in terms of genetics, we've sort of come up with a new metric for comparing specificity, or PRS. Imputation gives potential for improved power while retaining specificity. MTech does it in some cases, especially when there's family history data. And family history data is just worth a look in general, as it seems to be improving specificity the most, whatever method you throw at it. Finally, specificity is necessary for any cross-disorder methods. Otherwise, you will not be able to identify clear overlaps and use them to better your disease boundaries for improving those diagnostic criteria. Okay, so we're now on to the second part of the talk, which is to use genetics to assess symptom-disease relationships. And this is really much more about those symptom network things that I've shown you on 2 slides ago. And for depression, this is likely a very messy picture that is not going to be as simple as this here. It's probably going to be a huge mess with a lot of different symptoms and a lot of different networks because depression itself is highly heterogeneous, and I'll show you how they are. So there are numerous ways. At the moment, let's work with DSM criteria for now. I hope I've convinced you that that's the way to go for now. There are numerous ways to meet the DSM criteria for depression. There's 9 symptoms. You can have 5 of them, and then you'll be considered a case for depression. So different patients with depression may have completely non-overlapping symptoms. That's entirely possible. And they could also have very different onsets of disease at different points in their lives. There's early adolescent onset, peripartum onset for women, and some people have recurrent episodes much later into their lives. There are a lot of clinical subtypes that are defined in the DSM based on symptom criteria, as well as when they have onset. So these things are called DSM specifiers, and they're sort of like established clinical subtypes of depression that are in use right now. And you can't see anything in this study, but basically what they've done is taken all of the current DSM criteria specifiers for depression and asked whether they have the similar heritability or not. And they've shown that no, like they have different heritability. Okay. For example, one of those clinical subtypes is the atypical subtype of depression, which is consisting of this particular set of symptoms. And this study has shown that its genetics has got much more to do with BMI, triglycerides, etc., than schizophrenia, which is much more related to typical depression. That is not this kind of symptoms. Another example, there are some interesting studies on early onset depression having a much higher genetic overlap with schizophrenia and ADHD than later adolescent depression using PRS methods. The issue with this kind of analysis is that it's really hard to tell just by looking at them having different heritabilities and different genetic sharing with other things, whether any of them is closer to a natural kind. A lot of things can have different heritabilities from each other, and a lot of things can have different genetic sharing with other things. Whether those are necessarily getting us closer to the sort of network that we want to be calling a natural kind is hard to say. And relying on these clinical subtypes also completely prevents you from gaining any new insights into the sort of subtyping of depression. You're sort of confined to those definitions for now. So to really make breakthroughs in figuring out natural subtypes in depression, we have to start examining relationships between symptom and symptom networks with the disease. And since we're in the business of modeling things, we can start building simple models of symptom-disease relationships as a reference for us going forward. I'll explain what I mean by that. So for example, one simple model is where all the symptoms are consequences of the disease. And this is the disease. It's called psychological construct, not PC. And then these are the symptoms around it. So if, let's say, all of your symptoms are consequence of the disease, then when you want to look at heterogeneity within the disease, you should only look at within episode symptoms, like symptoms in people with the disease. Does that make sense? Because like people with different disease will have different symptoms. Okay. If that sounds too obvious to you, okay, let me show you why I even talk about it. There's another model that you can make, which is the symptoms cause the disease. In this case, if you want to look at heterogeneity of a disorder, you might want to look at symptoms in everyone, because people with different symptoms would constitute different subtypes of the disease, right? So these two are already quite different in terms of what you do in terms of identifying heterogeneity. And then finally, this is probably much closer to the truth, where symptoms cause each other, who knows what is going on, and then disease is somewhere around here. It's an emergent property of these kind of networks. So then this is kind of too difficult to think about, so let's not talk about that for now, but that's much more certainly closer to the truth. So what do people actually do nowadays? What are studies doing nowadays? They are mostly looking at the middle model, not because necessarily they've thought about all of these different kind of models, but mostly because N is large. Why? Because symptoms in everyone, you have more data on symptoms in everyone than you would have symptoms in depression cases, because cases are subset of everyone. That's by necessity, this is true. So what people have been doing is they are looking at symptom level investigations in UK Biobank, where they take symptoms in everyone. And symptoms in everyone looks like this. It's assessed with this thing called the Patient Health Questionnaire 9, where they ask, in the past 2 weeks, have you ever felt like you are depressed mood, have sleep problems, fatigue, etc. And the sample size is around 120K. So they've done a GWAS on individual symptoms collected in this way, and they've done genetic correlation of those symptoms with depression and a whole bunch of other phenotypes. More importantly, for subtyping, what they've done is they've done a structural equation modeling on the genetic covariance between these symptoms, and basically they're trying to cluster these symptoms based on their genetic covariance. And they've arrived at a two-factor model where they call these symptoms psychological symptoms and those symptoms somatic symptoms, and showing that they may be part of different networks. No, okay, you've sort of got the hang of this talk. We're going to start questioning this now. So is this model correct? Are we supposed to be looking at symptoms in everyone when we want to be talking about depression heterogeneity? Is this model the one that we want to be using? So one of my students have done some work assessing if symptoms in everyone equals to symptoms in depression in order to get to whether this model is correct or not. So firstly, what she's done is she's taken, she's calculated genetic correlation between symptoms in the general population and the MDD enriched population. What does MDD enriched population mean? In the UK Biobank, not only have they collected symptoms that in everyone for what have they been feeling in the past 2 weeks, they've also collected data on the worst episode symptoms in a slightly smaller data set of people. So here's genetic correlation across the 9 symptoms of depression between worst episode and current symptoms in everyone. And you can see that for most of them, it's not very clear here, but I can tell you most of them are not 1. So most of them are much lower than 1, actually. And that's not just because, like, when we assess current symptoms, sometimes we're not that stringent. So if we sort of, ramp up the severity criteria for the current symptoms, they are still not 1. So they are still genetically different, even if you require the current symptoms to be quite severe. We managed to recreate the two-factor model and sort of using the genetic covariance matrix of the current symptoms in the general population. So here is the same plot that I've shown you a few slides ago. And we realized the reason why it looks like this, where you have a two-factor model and the two factors are highly correlated with each other, is that the genetic correlation between all of these symptoms, the current symptoms that is assessed in everyone, are very similar to each other. They all have genetic correlation very close to 1. You can't see this that well in this projection, but they're all very close to 1. In comparison, the worst episode symptoms, their genetic correlations with each other are much lower than 1. So then when you do the same kind of structural equation modeling in using genetic covariance of worst episode symptoms, you get a totally different factor structure that has 3 factors, and they're not that correlated with each other. And this structure is much more similar to a lot of the modeling that has been done on the phenotypic level in family and twin studies. So what is the difference then between symptoms in everyone and symptoms within an episode of depression? And we can try to figure out what is contributing to the genetic difference between those two things. And one of them is, we've done genetic correlation of each set of symptoms with a bunch of non-depression traits. For example, subjective well-being, insomnia, you just have unable to sleep in your daily lives, whether you find there's meaning in life and life is not worth living. So they kind of correspond to some depressive symptoms, but they are feelings that you get as a person who doesn't necessarily have depression. And what we've seen is that the current symptoms that is assessed in everyone is almost always more genetically correlated with these non-depression phenotypes than the worst episode symptoms. Okay? We can also use Mendelian randomization, which is using genetic variants as instruments to test for causality between different phenotypes. We have the univariate model and the multivariate model. I'll explain it later on. But basically what you can do is that you can take the SNPs that are associated with some phenotype, let's say MDD or some non-MDD phenotypes, and ask if they are also associated with your symptoms. And a lot of assumptions sort of abiding, that would mean that this is likely causal. This sort of exposure is causal of this outcome. So using this kind of approach, we found that neuroticism, which is a personality trait, the genetic effects that are associated with neuroticism are much more often associated with the symptoms that is in everyone, as opposed to the symptoms that are only occurring in the worst episode. And that is true in the univariate models, where you test the genetic variants of neuroticism with individual symptoms, and as well as the multivariate model, where you test the same thing, but accounting for all of these other symptoms. So in other words, symptoms in everyone is not equal to symptoms in depression. They seem to be capturing a lot more of this general dysphoria, or sort of like general inability to feel happy, or general unhappiness in people that may not necessarily have anything to do with depression. So if symptoms in everyone is not equal to symptoms in MDD, is this model then still correct? And should we be using symptoms in everyone to look for depression heterogeneity? Maybe not. Maybe what's happening is that this is still true in depression itself, but then there's an outer circle of symptoms in everyone that is slightly distinct from these symptoms that are only happening in depression. So going forward, at least in my group, we will be using in depression symptoms to study depression heterogeneity. That's what we've learned from this exercise. And there are 2 projects that are ongoing that I would only briefly talk to you about today. You have to hear me talk again if you want to know what is going on with them. The first one is to try to identify etiological symptoms from the 9 that are currently part of the DSM. The idea is if among depression cases, between the genetic difference between the person with the symptom and without the symptom, if that genetic effect is also a genetic effect that contributes to depression itself, then that is etiological. Okay, I can use an analogy to talk about this a little bit better. So think about like drug effectiveness. So let's say I have two groups of patients. One is responsive to a drug and one is not responsive to a drug. I then look for the differences between those people who are responsive and those people who are not responsive. Now, there will be some drug responsiveness phenotypes where that genetic difference between people are also contributing to the disease itself. That's potentially where the drug, let's say, is targeting a particular pathway, and that's why they are responsive in some people and not responsive in other people. And that's where it's etiological. So then think of another case where if I look at that and that genetic effect is not part of the genetics that lead to the disease. That could be where the drug is, let's say, not metabolized properly. And the metabolism of the drug has nothing to do with the disease itself. So by looking at whether, the difference, by looking at the difference among cases in terms of a particular symptom or drug treatment response, etc., you can tell whether they are etiologically relevant to the disease itself. So we're testing it with this model here, and we're trying to figure out which of those 9 DSM symptoms are etiologically relevant to depression. So that's one project we have going on. Another project we have going on is to look at symptom-effect interactions. So this is based off a model called coordinated epistasis, which is basically saying that there's a lot of, each one of our symptoms is also extremely polygenic. So then there's a lot of genetic effects that are causing depression. These effects that are causing depression is probably made up of pathways that interact with each other, and then some of them are going to be captured by one symptom, and some of them are going to be captured by another symptom. So if we can model this and we can find those interaction effects, then we can know which symptoms need to work together in order to precipitate disease than others. Aside from all of that, there's another project that we've only started beginning to think about, which is how do general population symptoms relate to depression symptoms. And that's a whole other task that needs a whole other approach. I don't have time to go into today, but that's a whole thing that is interesting to think about also. So I've come to the end of part 2. So the part 2 summary is we need to be explicitly examining the underlying model of symptom level interactions for it to be informative in our analysis of depression heterogeneity. We need to think about what are we actually testing when we are looking at some results. And in terms of genetics, heritability and PRS predictions can't really tell us whether we're getting subtypes right. A lot of things would cause those things to be different. Factor analysis based on genetic covariances need to be performed in symptoms that are etiologically relevant to depression. Otherwise, I'm not sure what the factors are actually talking about. And then we have 2 more projects to come in the near future that would shed a little bit more light on the symptom and disease relationships. So some final takeaways from both of those parts that I've talked about today. Finding natural kinds is very hard. People haven't been able to do it for 110 years. How do we even hope that we do it within such a short period of time? But I think we now have quite a lot of tools that weren't available 110 years ago. And we can try our best, and hopefully we take some steps towards it. And it doesn't have to happen in our lifetimes, but if we take some steps towards it, maybe it will get there in the end. We can use genetics to figure it out, but only if we question the status quo. If we sort of don't take current definitions as it is, and we don't do studies just because the sample size is large, then we might be able to find things that we didn't know before. So I've come to the end of the talk. Thank you. These are my collaborators who have been instrumental in all of these studies that I've done. Also, this is my group. Simon here is at RECOMB. He is going to present something totally different from what I talked about. It's got nothing not related. And so please go to his poster at some point. Thank you for your attention.
Do you have any questions, or do we have time for questions? Yeah, I think we have time for some questions.
Okay. Sorry. Very very high level one, and I just thought about it. So the specificity plots, your phenotypes. Did people look into also omics or molecular match phenotypes in that data and try to help us verify? Maybe.
That's a good point. We haven't done it. It's possible to do it. That specificity matrix you can apply to any phenotype, and running it on different types of phenotypes will tell you different things. That's like not saying anything, but yeah, running it on molecular phenotypes, imaging phenotypes, etc., could tell you basically how those phenotypes relate to the phenotype that you care about, and that would tell you things.
Just sorry, I'm gonna cheat. Just following up on that. Yeah.
Like, if there's a way to do like a stratified correlation based on like cell type or something, that might more
totally. Absolutely.
Kind of like pointing in the right direction.
Oh,
maybe
totally.
have like a much better cell type consistency in the genetic correlation.
Yeah. No. Totally. Absolutely. We're thinking. So what you're saying is basically partition the genome into parts that are annotated different ways. That could be cell type specific expression, or pathways we derive from other polygenic methods. Or, for example, those things, what we've been thinking about is take those parts of the genome that are that we know have high local genetic correlation with a whole bunch of other things,
Right.
and those that with really low local genetic correlation with a whole bunch of other things. And if we did that, then we'd be able to basically know which parts of the genome are more specific to depression in their effects, and which parts are not. Yeah.
Thank you very much. It's a very interesting talk.
Thank you.
I was going to ask because I'm not super sure about UK biobank, but related to specificity, there is also another factor, misdiagnosis. It's also money in the water, so it's more.
Absolutely.
It's very high for mental disorders, especially for MDD. Have you checked these, or how do you try to incorporate this?
These are basically inherent in the data set that we have. So, yeah, they are. So, for example, if you had defined depression in, let's say, using the clinical way, we kind of know what the misdiagnosis rate is. It's quite high. But then, if you define depression using self-reports, that's going to be probably even higher. So we have a relative idea of how much misdiagnosis there are in the different types of depression that we have in our data set. And that's the whole bit about like which one of those are more specific to the clinically defined version. Now, what you're saying is clinically defined versions also have high misdiagnosis rate because of clinical practice, etc. That's the bit that we haven't been able to look at yet, because you kind of only can work with data that you have. But we are starting to collect. We have a consortium going on right now. We're starting to collect some data on people with electronic health record entries for depression, and then performing very detailed clinical interview on them to know what the rate of misdiagnosis is. And in picking those individuals to be part of this validation study, we're thinking of including individuals with, for example, 1 diagnosis of depression, 2 diagnoses of depression, diagnosis of depression and bipolar disorder, etc. So collecting a variety of things, and then asking in what circumstance is the misdiagnosis rate a bit lower. So using that, we should be able to sort of get a handle on misdiagnosis rates. Yeah, going forward, yeah.
Follow up a bit, because I think it's really nice also. I really like because the symptom-based approach related to that. But then it seems like this GWAS hits, especially like we are finding some hits related to some mental disorders. So how it's quite natural discussing this, but maybe it's difficult to say these are actually related to MDD, right?
Yeah. Yeah. So if you think about it, this is okay. So if you think about it this way, right? There are going to be some. So we've had like hundreds of GWAS hits now. There are going to be some hits that are correct, like that are relevant to depression and their effect sizes are correct. What are those hits likely to be? Those hits are more likely. Well, you can think about it various ways, but one of the ways that those hits will be correct for MDD is that they are not specific to MDD. They are highly shared between mental health disorders. and other things, such that their effects are the same across everything. Then whatever you get from a depression cohort, however misdiagnosed it is, that effect is still correct, right? Because like whatever else you throw in as depression is still going to give you exactly the same effect. Now that's why, you know, like when we were chatting about like partitioning genomes into pathways just now, that's kind of the thought behind it. If we could like identify those regions of the genome which is super shared across everything, then at those points your GWAS hits are fine because you can't get better. Those are the effects that you're going to get, whether you put in depression, pure depression, whatever that is, or other things. So those things are fine as it is. Other things are less fine because then their effect sizes for true depression, let's say, is quite different from their effects for other things that could be misdiagnosed as depression. So those things are harder, right? Those things you probably don't want to take those GWAS hits forward to anything. And then on the other end of the spectrum, there are those hits that are that only have an effect in depression and no effect in any other thing. That's pretty extreme, probably never going to happen. But let's say there are those hits, then your GWAS is again totally fine, because whatever else you throw in is just noise, right? Like it doesn't do anything. Your effects are still, it's going to be slightly less precise because it's like basically effectively reducing your sample size, but effects is still the same. So then if you start thinking about stratifying GWAS hits this way, then you can already, from those hundreds of hits, start to know what is usable and what is not usable. And then you can start doing like additional analysis based on that.
Nice talk. And mine is more comment on myself rather than questions. So when you talk about this natural kind, right, or trying to cluster in symptom into what's the real disease, I'm thinking, what do we mean by that? I think it's, or what's the purpose of that, right? I think all this DSM is really for clinicians so that they can treat the patient. You give it nice, precise diagnosis so they can treat the patient and manage the goal. So I guess our goal here is also trying to find the type of symptoms or type of disease so that certain drugs or whatever the treatment will work for this group of people,
Absolutely.
right? So that's the final goal.
Yes.
So if you go from there, then symptom by symptoms like depression, it may not even be that much relevant. I mean, some relevance. So like you were talking about using the genetics that could help. So from there, it's almost like if certain drugs work on certain pathways, then if we can find these pathways or genetics included, using this to define whatever disease we call it, whether it's a major depression or it's a part of the bipolar, if certain drugs can work on this group through this pathway, then you can define that as a disease, right? So almost you can do things like, because that's, if the final purpose is to find a treatment for this group, because the problem with all this heterogeneity is that you treat people with the same standard of care method, but some work, some don't. It's probably because of classification. Classification is so broad and all that.
Yeah.
So I think, going from genetics to symptoms, I think it makes more sense.
I see. So I think what you're saying is complementary to what we're doing now. Looking at drug responses is actually the next thing that we are hoping to do with sort of now the increasing sort of availability, I guess, of electronic health record data. Whereas, that's where we get most of the prescriptions and sort of treatment intervals and whether people stop taking particular drugs, etc. Using those information, we can start deriving phenotypes that are more related to how effective a particular drug is, and using that we can then, for example, like you said, use the genetics of that to try to figure out what those pathways are.
And the other thing we're showing earlier, using different symptoms to impute the MDB, and then you get different powers or different hits for the GWAS. And like one example you gave was using the family history, the other was more of a environmental stress and all that. So that's almost like you're really looking at different pathways to what is the so-called major depression, right? So it's like one pathway is through this, okay, so they're all genetics, so I shouldn't say one is through inheritance, one is through others, but one is like through this depression-like symptoms, inheritance, running through the family. The other is the like stress, response to stress or response to trauma, then that causes depression. So that perhaps is a different pathway. So as a result, you end up with different sets of GWAS, which might be indicating their different type of disease, right?
Absolutely. So that's another thing that we are hoping to do, like expand into later on. So for example, for MTAG and also for all those imputation methods, one thought that we haven't been able to do is what if we started including lots more things? Also like what Simon said just now, what if we started including molecular phenotypes and imaging phenotypes, like brain imaging, like metabolomes and all of those kind of things. Then, and also, environmental factors, family history, like you said. If we started including all of those, then you see that for some of those types of data, they would help imputation more than others for certain things. And some of those phenotypes are going to help, this is a nice way to test whether it's biological or not. It would help across cohorts. So for example, it helps in this cohort, it also helps in that cohort, and it also helps in this other ancestry, and etc. So then let's say you find those phenotypes that helps across a lot of different things. And those are much more likely to be biological, and those are much more likely to matter to the disease itself. So one of the goals is actually to find out what those are, and then using those as clues as to what is going on in the disease.
I guess when people use this, all the symptom spectrum from everybody, the entire world, and the probability to find are those things. So the whole.
Yeah, we can take this, offline. Yeah.
We're going to have time, maybe.
Yeah, I think. If we have any other questions, maybe let's move them offline. Let's think again.